So last week, we ended by basically introducing a bunch of public health and epidemiology related problems that we want to try to solve. And basically each week, starting with today all the way till week 8, because we only have 8 weeks in this class, we're going to be presenting a different technique or a different computational or bioinformatics problem that addresses each of those problems. So today what we're going to do is put ourselves in the shoes of the very first researchers who are trying to figure out what is this novel pathogen that's infecting people in Wuhan. So our topic is going to be, how do we sequence the first viral genome of a novel epidemic? So today's goals, I'm going to talk a little bit about DNA sequencing. So we're going to cover a very, very high level description of how DNA sequencing technologies work. We're going to talk about genome assembly. We're going to do some terminology about genome assembly. So contigs and scaffolds, those won't make sense to you yet, but hopefully later in the lecture, they'll make a little bit more sense. And then we're going to end with, how do we assess how good a genome that we've assembled actually is? So the first topic is DNA sequencing. And you might be wondering, well, why are we talking about DNA sequencing if this is an RNA virus? I'll get to that in a little bit. But yeah, so first, some context. I'm going to explain how DNA sequencing works and kind of the computational problem we have at hand by using an analogy. So I'm going to start with what I call the newspaper problem. So imagine that I have a bunch of stacks of the exact same newspaper, so a bunch of identical copies. And then imagine that I put a bunch of dynamite under those newspapers and I light the fuse. And of course, don't try this at home. This is only for computer science professors to do. And then kaboom, the dynamite explodes, all of the newspapers are blown to shreds, and we have all of these tiny charred fragments. And the computational question that I want to ask is, using these tiny burnt up fragments of multiple copies of the exact same newspaper, can I somehow reconstruct what the original newspaper said? And if you think about it, you might realize, okay, well, one approach that I should have is, if I take two fragments of that newspaper and I look for overlaps between them, I can kind of layer on top of each other the different overlaps and try to piece by piece reconstruct the newspaper. So how is this related to DNA sequencing at all? Well, basically, we have a bunch of copies of our newspaper. All of these copies are identical. It's a similar process with DNA sequencing, where our newspaper is the genome of whatever that we're trying to sequence. So we have a bunch of identical copies of the viral genome. And the DNA sequencing technology, for reasons that you don't have to understand yet, but hopefully later in like a molecular biology class or something you'll dive into. But basically, the DNA sequencing technology randomly shatters the DNA into all these tiny fragments. And then we're able to basically read out a subset of those tiny fragments. So why that is, don't worry about, just take my word for it that we're only able to reconstruct relatively short fragments, typically between 100 to 150 letters long. Whereas for COVID-19, the genome is like 30,000 letters. So yeah, we only get a subset of our complete short strings. We only get a subset of those small fragments. And then that was DNA sequencing. With RNA sequencing, it's essentially the same process, except we first convert our RNA into DNA via a process called reverse transcription. And then we just run it through DNA sequencing as normal. So for COVID-19 is an RNA, or SARS-CoV-2 is an RNA virus, but we just reverse transcribe it into DNA, and then we do DNA sequencing on that DNA that we've created. So yeah, any questions about how DNA sequencing as a concept works? So basically, I have a bunch of copies of my viral genome. I stick it in the DNA sequencer, and then I get basically fragmented chunks of that genome, and I can read out short fragments that are subsamples. So I don't get to see every single short fragment. I get to see just some of the short fragments. There's a question, shouldn't there be more identical sequences? Yeah, so here I just, for the sake of fitting it on a slide, I only put like 4 copies of the genome. In reality, there's going to be millions of copies of the genome. So you're creating millions of copies of the genome and then fragmenting them and then randomly picking out short fragments. Yeah, great insight. So it wouldn't just be 4 copies, it would be like millions. Yeah, any other questions about that? Does that kind of make sense? And if people are interested in learning, like, how do these technologies actually work in terms of the molecular biology, feel free to email me and I can maybe share some resources on how these sequencing technologies work. Cool. Okay. So where are we at now? Basically, what we've done is we've created all of these tiny fragments of our genome. So the COVID-19 genome happens to be, or SARS-CoV-2, the SARS-CoV-2 genome happens to be roughly 30,000 letters long. And I've now been able to read out roughly, millions of these short 100 to 150 letter long fragments. And my computational problem now is, how do I take these short fragments and piece them together, much like we did with the newspaper, in order to reconstruct the original entire genome? So this is a computational problem known as genome assembly. Basically, we're trying to assemble those tiny fragments into the complete genome. And yeah, just like with our original newspaper problem, what we're doing is looking for overlaps between our fragments. So here we have this red fragment, we have this green fragment, and we have this blue fragment. And you'll notice that they have this relationship in terms of their overlaps. So I can kind of piece them together by saying, oh, well, the suffix of this green fragment is identical to the prefix of this blue fragment. So maybe it was green fragment and then blue fragment came immediately after it. So yeah, we try to use these overlaps to reconstruct the genome. So any questions about what our bioinformatics problem is? The input and the output. So input is all these short fragments. Output is the single genome sequence. Okay, so the general approach to solving this is by using what's called a Bruyne graph. So let's pretend that I have every single possible short fragment. So let's say that my genome was length, some length, and let's say that I'm able to read short fragments of length 3. And for the purposes of this simple example, let's assume that I have every single possible substring of length 3 for my original genome. So in reality, I told you the DNA sequencer is going to lose some of them, but let's just pretend that this is every single possible fragment of length 3. So a De Bruyne graph, basically what we do is first we create one node for each unique prefix and each unique suffix, like basically each unique prefix or suffix of the reads. And then for each of our reads, and a read is just one of these fragments, for each of our reads, we basically place an edge pointing from the prefix node to the suffix node. So I'm going to show you how that looks like with this example. So I'm going to look at the first read, and I see that the prefix is AC. So I create a new node for the 2-letter long string AC. And then the suffix is CA. So I create a new node labeled by CA. And then I put an edge from AC to CA. So from the prefix to the suffix. Okay, everyone with me so far? Any questions about that first one? Kind of makes sense? Okay, I see a thumbs up, perfect. Let's go to the next one. So the next one is ATT. So I create a new node for AT because it doesn't already exist. And then the suffix is TT. So I create a new node for TT. And then the entire read ATT, I'm representing as an edge, right? This link from AT to TT. Okay, so what about the next one? So what do I do? So now I'm looking at GAT. What's the first thing that I have to do? Feel free to type in the chat. Feel free to unmute. So if I'm handling GAT, what do I have to do? Create a node with GA, exactly. So I see the prefix is GA. I don't already have a node labeled by that, so now let me create one. And then for this one, you'll notice the suffix AT, I already have a node. So I mentioned I only create nodes for unique prefixes and suffixes. So I don't want any copies. If I've already seen a given prefix or a suffix, I don't want to create a new node for that. So I only created a new node for GA. And then I'm just reusing the node that I already created for AT. Okay, so now what do I do? So I have a node for the prefix. I have a node for the suffix. Now what do I do with this read? Feel free to unmute, feel free to raise your hand, feel free to type in the chat. Any thoughts? So what do I do? So I've created the node for the prefix, I've created the node for the suffix, and then yeah, exactly. I would connect GA to AT with an edge labeled by GAT. Exactly. I create an edge from the prefix to the suffix, labeled by that read. Okay. So then next, TA is the prefix. I don't already have it, so I create a new node. AC is the suffix, but I already created a node labeled by AC, so I don't want to make a duplicate. And then TAC is going to be an edge going from TA to AC, and it's going to be labeled by that read, labeled by TAC. And then lastly, TT already exists, TA already exists. TTA is going to be an edge from TT to TA. And this is what we get. We call this network, we call this a de Bruyne graph. So how does this help us? I just did all this super complicated stuff. How does this help us reconstruct the genome? So does anyone see any patterns? Does anyone want to kind of venture a guess? How can I use this de Bruyne graph to reconstruct what the genome might have been? Feel free to unmute. Just speculation. What do you think? Like, does anyone see any patterns here? Anything that might help us?
I'm still trying to figure out. So when you went from AT to TT, and then you went from TT.
Okay. Oh, sorry. I'll just keep them all there. Yeah. So when I went AT to TT, yeah.
And then you have TTA. Could you have gone from the TA to like the AT above the TT?
No, so TTA, it has to be from the prefix to the suffix. So we have to go from the beginning to the end.
Okay.
Does that kind of make sense? So yeah, so TTA has to be TT. Oh, sorry. It has to be, wait, oh, why is, yeah, it has to be TT to TA. To be TTA.
Got it.
Kind of make sense?
Yeah.
Yeah, this one, it can be tricky.
Okay. And then from the TA, if you have like TA, you couldn't do like TAT.
So if we wanted TA to go to AT, we would have needed one of these reads to be TAT, so that the prefix would be TA, the suffix would be AT. So it's purely because we just happen to not have that.
Okay.
Does that kind of make sense? But yeah, exactly. If I had TAT, that would mean from TA to AT.
Okay.
Nice. Yeah, so someone mentioned in the chat, so maybe write down the sequence in the arrow form to get the sequence. So yeah, so someone mentioned, maybe I can use these arrows to reconstruct the genome. Okay, and then someone guessed GAT. Okay, so someone said GATATT, and then someone said GATTACA. So how did you get the second one? How did you get GATTACA? Do you want to walk me through that?
Well, I started at GA and I went to AT, but when we did that, the A is overlapping, so I didn't count it twice.
Okay. Yeah, exactly. Yeah, so that's the right answer. So let's work through that. So basically, I think the pattern that the student recognized is, hold up, well, it looks like this is the only node here that doesn't have anything going into it. And it looks like this one over here is the only node that doesn't have anything coming out of it. So what if I just started here and just kind of jumped over these arrows one by one and try to reconstruct the genome. And remember, I told you that the reads, let me see if I can go back to that slide. Remember that I told you that the reads overlap with each other. So like if I have this read here, this green one, and then I have this blue one, in the original genome, they overlap, right? So this is the window between the green one and the blue one. And basically the only new letter that the blue one provides that the green one didn't already have is this last letter. All of these other letters overlapped with this green one already. So I already counted it if I counted this green one. So using that exact same logic, we can look at the overlaps in this De Bruyne graph. So I can basically start at the beginning and I would say, okay, so the very first edge that I traverse, the very first arrow that I go across is GAT. So I'm going to say that's the first 3 letters of my genome. But then every subsequent arrow, basically all of the letters at the beginning, so all the letters except for the last letter overlap, right? So here, ATT, well, that AT in my original genome, the AT is already counted, right? It's already counted here. So let me just, as I go through all the subsequent, whoops, why is it not clearing? There you go, clear all my drawings. So for the first one, I counted all of it because that's the first one that I've seen. But for all the other ones, I'm only adding the last letter to it. So if I look at the next one, ATT, I'm only adding this T at the end because it's ATT. I've already counted the A and the T at the beginning. So does that kind of make sense so far? Feel free to jump in. It's weird, but yeah, it makes sense. Okay, so then what do I do next? So what's the next one? Feel free to type in the chat. Feel free to unmute. Got to make sure everyone's still alive.
Is it just A because you already have TT?
Exactly. Yeah. The next edge is TTA, but as you said, I already have the TT, so I just add the A at the end.
Oh, and then you would just add the C and the A?
Perfect. Yep. Exactly. Okay. And then now I don't have any more arrows that I can go by. So I'm done. And that is my genome sequence. And if you haven't seen it, this is actually a movie. It's called Gattaca. It's really good. So check it out. So yeah, so in reality, it gets a little bit more complicated than this because as I mentioned, maybe some of these reads got blown up. Maybe, the sequencing machines themselves, they make mistakes. They're not perfect. So maybe you have some mistakes in your reads. But for the purposes of our class, don't worry about those. Just know that there exist really, really smart people. So someone mentioned that they're from Moscow. So this is actually from St. Petersburg State University. So, I don't think those are actually necessarily very close by, but also in Russia. So this is from Pavel Pevzner's lab at St. Petersburg State University. And it's a tool called Spades that uses this approach, this De Bruyne graph approach, but also has some clever tricks to account for any of those real world problems to do this genome reconstruction. And this is actually one of the best practice tools used for doing viral and bacterial genome assembly. So in the future, you yourself are never going to have to do this. This is a pain in the butt. But what you can do is just take this existing tool and run it and just know that behind the scenes, this is kind of the general approach that it's taking. Does that kind of make sense? So basically what we would do if we were the very first epidemiology researchers studying this virus is we would take our viral sample, we would stick it in the DNA sequencing machine, and then that's going to spit out all of those short fragments. And then we're going to take those short fragments and just stick them into Spades. And Spades is going to do all that crazy computation and output a genome sequence, the Genome Assembly. Okay, so everyone cool up to this point? Any questions so far? Feel free to unmute. Feel free to, I see thumbs up, perfect. Okay, so now I'm going to introduce some terminology called contigs and scaffolds, and these have to do with the genome assembly. So as I mentioned, it very well could be that some of those fragments that we need, some of the parts of the genome that we were trying to sequence, we might just have never seen because they blew up. So in our newspaper, we might have just blown up pieces of the newspaper and are unable to reconstruct them. So a contig is basically a contiguous segment of the genome that it's able to assemble. So maybe the true genome was like this entire length over here, but maybe this segment over here got blown up completely. The sequencing machine wasn't able to read any of it. And then this segment over here was also blown up completely. So the sequencing machine wasn't able to read that either. So when we run SPADES, maybe SPADES is able to reconstruct this part of the genome and this part of the genome and this part of the genome. But because the sequencing machine totally blew up this part and this part, it just doesn't even know that those exist. So we call these contiguous pieces of genome that our assembler is able to assemble contigs. For contiguous, we say that they're contigs. So the genome assembly that we actually get, it might not be a single cohesive sequence. It might be a bunch of chunks called contigs. And then if the genome assembler is able to figure out the order of these contigs, so maybe the genome assembler is able to figure out that this is the first contig in the genome, and then it's followed by this second one, and that's followed by this third one. If it's able to determine the order of the contigs, we call that a scaffold. What these words mean, don't worry about too much, but basically just know that we might not be able to reconstruct the entire sequence. We might only be able to reconstruct these chunks of it. Does that kind of make sense? Ish? Yeah, okay. Okay, so let's say we run our genome assembly, we get those contigs, we get those fragments of contiguous genome that it was able to assemble. How do we determine if our genome assembly was good? So let's imagine that I run SPADES and I get these contigs here. So this is the first contiguous sequence that it was able to reconstruct. This was the second one, this was the third one, and here I have them ordered in decreasing order of length. So really all of these came from a single genome, but these are all just each like little chunks of the genome. So like, let's say this one is, how do I do my colors? Let's say this is orange. So let's say that's from that part of the genome. And let's say that this one is like from this part of the genome. So really all of these came from a single genome, but we were only able to reconstruct chunks of it. How do I determine if I did well? Like, did I do a good job? Yes or no? We have to define some type of metrics that can help us measure how good we did. And what we're going to do is define a metric called N50. So I'm going to define the total contig length to be the total length of all of, so total contig length of T, and this part's going to get a little bit mathy, so try to bear with me. So I'm going to define total contig length of T as the total length of all of the contigs that are at least T letters long. So basically, if you tell me total contig length of 100, for example, what I'm going to do is count all of the contigs that have length greater than or equal to 100. I'm going to add up all of their lengths. So the N50 metric, this is one of the metrics that we use to determine how good we did. It's the longest possible length. So it's the maximal contig length. It's the longest possible length such that the total contig length of that length, so the sum of all of the contigs greater than or equal to that length, is at least half of the total length of the contigs. So this is really, really weird. I'm going to show you it with an example. So let's put some numbers on here. Let's say that this contig is 70 letters long. Let's say that this contig is 60 letters long. Let's say that this contig is 30 letters long. This one's 20 letters. This one's 10 letters. So the total length of all the contigs in this example is 70 plus 60 plus 30 plus 20 plus 10. So 190 is the total length of all the contigs. I just added up all their lengths. People with me so far? Okay. So N50, I said, I'm looking for a contig length such that the sum of all the lengths greater than or equal to that is at least 50% of the total length of all my contigs. So first of all, 50% of the total length of all my contigs. So 50% of the total length of all my contigs. Total length of all my contigs was 190. 50% of that is 95. Okay, so people still with me? So I added up all their lengths divided by 2. Oh, someone asked in the chat, can you please explain again what a contig is? It's a part of a genome sequence? Yes. So a contig, each of these is a contig. It's just a contiguous segment of the genome. We weren't able to reconstruct it all in one go, kind of like if you're like ripping off a sticker. We weren't able to take it all off in one go. We were able to take it off in little fragments. So that's what a contig is. But yeah, so 50% of the total length of all of them is 95, right? 50% of 190. So what we need to do now is see, okay, so if I just look at the single longest contig, so if I look at the contig that has length 70, the total contig length of 70, so the sum of the lengths of all of the contigs at least length 70, well, there's only one contig that has a length greater than or equal to 70. So the total length, the sum of all of their lengths, well, there's only one, its length is 70. So 70 is less than 95. So I'm not done yet, right? Because I'm looking for at least 50%. I'm looking for greater than or equal to 50% of my total length. So at least 95, but 70 is less than 95. So I'm not done yet. So let's try looking at the next longest contig. So if I'm looking at 60, total contig length of 60, well, this is the total length of all the contigs that have a length at least 60. So that means it's the length of this contig has at least length 60, and this contig has length at least 60. So the total length of all the contigs that have a length of at least 60 is 70 plus 60, which is 130. 130 is greater than or equal to 95. So 130 is at least 50% of my total length. Therefore, I say that the N50 of this genome assembly here is 60. Okay, so I'm just going to pause a little bit. That's a lot of numbers that I just threw at you. So we're going to try to internalize what this means. So if you think about it, which do you think is better? A big N50 or a small N50? Do I want this number to be big or do I want this number to be small? So maybe think about that for a little bit. It's not necessarily obvious because this is a really weird concept. But do you think good means this number is big? Or do you think good means this number is small? Maybe think about that for a little bit and then we'll discuss. And try to think of the extremes. Try to think of what is the best possible scenario for your genome assembly. Okay. Does anyone have any thoughts? Feel free to unmute. So do I want this to be big or do I want it to be small? Okay, so okay, go for it. Yeah.
I wasn't entirely sure, but I guess I would kind of vote for bigger just because you want to find out more information about like the bases, like AGT, to like piece together your genome that you're looking for.
Mm-hmm. So what is the best possible scenario for my genome assembly? So like, let's say that my let's say that 190 really is the length of my genome. So let's say my true genome actually is 190 letters long. And in this example, I wasn't able to reconstruct it in one go. So like, imagine it's a sticker that I'm trying to peel off. It kept breaking, but let's imagine the best case scenario is that I was able to assemble it entirely. So I was able to rip it off in one go. So what's the best possible scenario? Best possible scenario is that I sequenced it in one go. So I got the entire genome in a single contig. So you're actually correct, but how does that look like? So let's say that my genome, let me actually move this number, I put it in the wrong spot. So let's say my genome actually is 190 letters long. And let's say that my genome assembly was the best possible. The best possible means I was able to reconstruct the entire genome from start to end in a single contiguous sequence. So my genome assembly would be one contig that has the length of the entire genome. So one contig of length 190. This is the best possible scenario. So what would the N50 look like in that scenario?
Would it be like N50 is equal to 95?
Would it be 95? So basically, remember, it's the length of the contig such that if you add up all of the contig lengths greater than or equal to that, it is at least 95. So we're still comparing against 95, but remember, we're looking for the length of a specific contig. So here, the N50 was 60 because the contig that has length 60 and everything that's larger than that, if we add those up, it became greater than or equal to 95. Yeah, so someone mentioned in the chat, 190, exactly. So if I had that scenario where I had a single contig of length 190, if I went through these steps exactly as I did last time, I would say, okay, total contig length of 190, that is the total length of all the contigs that have length at least 190. Well, only one contig has length at least 190. I only have one contig. So the total contig length is 190. 190 is greater than 95. Therefore, that length is my N50. So it's kind of weird, but in this optimal example, 190 is actually our N50. It's the length of the longest contig such that it's the length of, it's the largest length such that if you added up all the contigs greater than or equal to that length, it would be more than 95. But there's only one contig. So it itself, yeah, so if that's not super clear, that's totally fine. This thing is a really weird metric, but my main point that I was hopefully trying to make is just, we want N50 to be big. Bigger N50 is good. That means that we have more of our genome in long contiguous chunks. Does that kind of make sense? It's not super intuitive, but I hope once you think about it a little bit, yeah, we want N50 to be big because that means more of our genome is in just a couple long contiguous chunks. Does that kind of make sense? Feel free to unmute. Feel free to type in the chat if you have any questions. And it's really weird. I totally feel it for you, but we're gonna get to a simplification in a little bit. Okay. Oh yeah, go for it. Was it? Oh no. Okay. So yeah. So N50, we were looking at the total length. So what we were comparing against was, so the 50 here is this 50, and what we were comparing against was the total length of all the contigs. So what we were comparing against was, we added up the lengths of each of these contigs and got 190. And then the 50 meant 50% of that. So this is useful if I have no idea about anything about whatever virus I'm looking at. But what if I somehow knew the true length of the genome? So what if someone magically told me, hey, COVID-19 has a genome length of 29,000. And there are like biochemical techniques that we can do to estimate the length of a genome without necessarily sequencing it. There's a similar metric called NG50, G for genome, where if we do know the length of the genome, it's the exact same metric, except instead of doing 50% of the total contig length, I do 50% of that genome length that you somehow magically told me. So let's pretend that I magically know that in this example, so these lengths are the exact same. But let's say that magically you told me that the total length of the genome is actually 270. So in the previous example, I was just adding these up, but now I know that the genome length is actually 270. I do the exact same thing. So 50% of 270 is 135. Okay, so I look at, I'm going to just jump forward to here because we already worked through the first example, but yeah, let's jump straight to here. So total contig length of 60. So the total length of all the contigs with length greater than or equal to 60, that's 60 plus 70, which is 130. This is less than 135. So I'm not done yet. I need it to be at least 135. So I'm not done yet. Let me look at one more contig. So total contig length 30. So I took into account the next longest one. Total contig length 30 is the sum of all of the contig lengths greater than or equal to 30. So that's 70 plus 60 plus 30. That equals 160. That is greater than or equal to, so that is at least half the genome length. So that tells me that my NG50 is 30. It's the length of this contig, because if I take that contig and everything greater than it, I get at least half the genome length. Okay. Everyone kind of follows-ish. And then just like with N50, I want this number to be big. The bigger this number, the more of the genome I have in kind of single, really long contigs. Okay, so the last thing that I want to show you is we can make a plot that looks like this. And so kind of the icing on the cake here is all of this really annoying, confusing math that we did. You don't have to worry about it. There's a tool called Quas, which is also from that same lab at St. Petersburg State University, that it does all of that metric computation for you. So what you do is Spades, the genome assembler, gave you a genome assembly as its output. You just plug that into Quas, and Quas does all of these weird genome accuracy metrics for you. But one really useful thing that Quas outputs is a plot that looks like this. So on this plot, what I'm doing is I'm sorting each of the contigs in decreasing order of length, just like I did over here. And then starting with the longest one, the length of that contig is the y-axis. And what percent of the total contig length I've seen so far is the horizontal axis. So basically, I'm just kind of overlaying each of those contigs on a plot. So the first contig that I had length 70, and that was like just under 40% of my total genome, like my total contig length. Then the second contig that I had was length 60, right? So I put that at y equals 60, and it is this range of my total length. So up to this point, this was roughly 70-ish% of my total contig length, composed of just these two. And then the next longest contig had length 30, the next one had length 20, and the next one had length 10. And Quast will do this for you. Okay, so again, just kind of stepping back, let's think of the extremes for this plot. Maybe this plot, one extreme is that it looks like this, and one extreme is that it looks like this. Or actually, maybe I'll just do like this. Oops. This. Which of these two do you think is better? Do we want it to be this red looking one where it's big pretty much all the way through? Or do we want this, actually it wouldn't even have, I don't think it would even drop. I think it would just stay constant. Or do we want this purple one where it stays low all the way through? Which do you think is the better situation? The red one? Yeah, exactly. So this upper one is the better situation, because if you think about it, the best possible example is instead of having all of these tiny little fragments, the best possible scenario for my genome assembler is if it's able to sequence the entire genome in one long contig. So what would that look like on this plot? It would be basically just a single long line that is 100% of the total length, and the vertical axis would be the length of that contig. So we would want a really big, long contig is the best possible scenario. So again, you will never have to construct these yourselves, even if you're doing research in this space. You're just going to plug it into this tool, and this tool is going to compute the N50, the NG50, it'll compute this plot. The only thing that I want to make sure that people are able to take away is if you do this, so if you create a genome assembly, plug it into this tool, I want you to recognize that we want N50 to be big, we want NG50 to be big, and we want this plot to be big. Those are the main takeaways. Okay. So I'm going to end this lecture by saying, okay, what do we do with this? So let's assume that I did my DNA sequencing experiment. I have my viral genome assembly. I've done my quality checking using this tool Quas, and I've determined that, yes, I think my genome assembly is good. What do I do with that information? Well, there's a tool called BLAST, which you can think of BLAST kind of like the Google search of biological sequences. You basically type in a sequence that you're interested in, and BLAST has this big database of existing sequences that it already knows about, and it looks up which sequences in the database are most similar to the sequence that you looked up. So if we take the COVID-19, so the SARS-CoV-2 genome assembly, and we search it against BLAST, and in this example, I've limited it to only include sequences that existed before 2020, just to put ourselves in the shoes of those researchers. So I took my genome assembly, plugged it into BLAST and said, hey, BLAST, tell me what does this look like? You'll notice that the top hits are bat SARS-like coronaviruses and actual SARS coronavirus. So now this tells you without any kind of medical expertise, all you needed was the genome sequence. This starts telling you, hey, I think we might have a virus that's similar to SARS on our hands. Does that kind of make sense? So we can just kind of plug it into BLAST, which is basically the Google search of sequences, and figure out what does this look like, and that tells us what we might be dealing with. Okay, so that's everything that I had. So I'm going to maybe pause a little bit. Are there any questions? Does that kind of make sense? Feel free to like chime in. Any questions, even if it's not related to this? So the basic takeaway of today's lecture was we take our unknown viral sample collected from a person, we run it through a sequencer, we run the output, like those reads that we get, into a genome assembler, we run the genome assembly through this quality assessment tool, and then we also run our genome through this database, and we can find which existing viral genomes look like the one that we've just collected.